First of all, I would like to start thanking all the attendees to this CLASSY Scientific Symposium. Very briefly, in even less than 5 minutes, I will try to put this into context. So basically, this is just probably one of the last events of a FET Open European project that put together various academic groups and also some companies. And basically, we have been working on developing new concepts of catalysis from the systems chemistry perspective. So trying to use chemical networks, supramolecular assemblies, and let's say to disseminate our results, but also to engage with experts in the field of systems chemistry. We have put together this symposium, which I hope it will be useful. So I would like to thank the external invited speakers. They are of a very high level. So we have today with us Professors Dora Tang, Silvia Marchesan, Rink Elkema, and Leonard Prince, and their talks will be intercalated with results from the CLASSY network.
Good. So yeah, thanks for the introduction. I'm going to jump straight into discussing our results on catalytic templated oligomerization. Inspired by nature, chemists have created both DNA-based as well as fully synthetic templates, which then allowed them to prepare complementary DNA and RNA strands as well as other types of linear, cyclic, and even topologically more complex oligomers from the monomeric building blocks in a length and sequence controlled way. Thus, we can safely say that templated synthesis is a powerful and enabling methodology to access oligomers from monomeric building blocks in a controlled one-pot process. I hope I was able to show you that we created synthetic templates which can construct linear oligomers in a length-controlled manner in a catalytic process. We can control the length of the formed product by choosing the size of the template and the number of its activation sites. The oligomerization is achieved by a combination of recognition and activation events which facilitate the process. And we hope that these results open exciting possibilities for the development of future supramolecular catalysts for controlled assembly of complex molecules from simple individual components. Finally, I would like to thank a few people. I would like to thank Helma Venemers for being able to work with her on this exciting project. These four gentlemen, Dario, Dominic, Rudiger, and Martin, whose hard work and ideas contributed to this project and without whom we wouldn't have been able to achieve these results. Or the other members of our CLASSY team at ETH Zurich, Alena, Jasper, Jonas, and Leo. All the other members of the Venemers group, of the whole Venemers group, for creating a wonderful atmosphere to do science. All the CLASSY partners, it was wonderful to be part of this consortium and work with so many great scientists over these last few years. The European Union and the ETH Zurich for funding, and all of you for kind attention. I'm happy to take questions now.
Thank you. Nice discussion, but it's time to go to the next speaker. Pleasure for me to introduce now Professor Rink Elkema. He comes from the Technical University of Delft, and he's actually a great expert in systems chemistry and application into dynamic materials. And today he will tell us about chemical reaction networks based on conjugate additions on beta-substituted Michael acceptors. So, Rink, the screen is yours.
Thanks very much for the invitation. It's a very interesting conference for me. So, what I would like to discuss with you is, well, we've been doing a lot of work on chemical reaction networks over the years, and in the last few years we've mainly focused on one specific type of reagent in this, a beta-substituted Michael acceptor, which offers some quite unique possibilities there, and this is what my talk will be about. So we created a new network starting from a proline-based Michael acceptor reacting with a thiol, and then in the presence of an oxidant you go through from the starting material to the thioether, then you oxidize that thioether first to the sulfoxide and then to the sulfone, and then it's a good enough leaving group to go back to the starting point. So you close the cycle. Then we found out that depending on the strength of the oxidant, you can get many different paths in such a cycle. So the original one is the one here that uses a strong oxidant because that's what we need to get to the sulfone. But if you offer it only a medium strength oxidant, then you can only get to the sulfoxide. And then the reversibility in the system allows it to even go from there back to the starting point, proline atom. If we offer it even a weaker oxidant, then the sulfur oxidation is out, but it can still go back simply by reversing the reaction. And then it turns out that the weak oxidant can oxidize the thiol to the disulfide. That pulls it from the equilibrium, and then you drive it back to the starting point. So with the oxidant, we can address many different paths in such a network, and thereby create these different shunts. And what we also found out here is that these are all forward processes, of course, but by introducing, for instance, a reductant such as TSEP, so a phosphine, we can have the large amount of disulfide that is created turned back into a thiol, and that then starts to drive back some of these reactions. So we can even we can both control paths, but we can also drive certain paths back in the opposite direction with the redox chemistry that is involved with this cycle.
Well, then I thank you again for sharing your results with us, and now let's move to the next speaker. So he comes from our consortium from CLASSY. He's Professor Wolfgang Krautil from the University of Graz, now more in the biocatalysis side of the consortium, I would say, and he will tell us about multi-biocatalytic reaction systems. So Wolfgang, now is your turn.
Thank you very much, Andres, for the introduction, and I'm happy to be able to speak today here. So, well, when we move now to biocatalysis and say what is about reaction systems, well, there's also these buzzwords in biocatalysis like systems biocatalysis, biocatalytic reaction systems. What we understand here is mainly that we go for a cascade and choose and design a cascade. So not look what nature is doing, but design our own cascade and choose for this cascade the best suitable enzymes, and then try to make this very efficient. And in the consortium, as I will show you, we try to extend to get this to organocatalysts, combine enzymes also with organocatalysts for the coupling. Well, what was the important part was when we checked for the enzyme, we noticed another narrow operational window. For the peptide, the operational window was clearly different, but we tried to make a compromise. And what was finally the important point is that we could have engineered the enzyme, but the group of Helma Wennemers and Jasper Möller modified the catalyst, actually the peptide catalyst, to make it more efficient in the aqueous solution and more better compatible with the enzymatic reaction. So what was finally then shown is that a number of substrates could be transformed, and while getting the products out with actually rather good yield and with EEs up to 98% here. And well, with that, I would like to acknowledge the funding and of course the two cooperation partners in these projects I showed here, Wilhelm and Helma, and of course Miguel, Jasper, and all people involved, and of course the whole CLASSY consortium for the excellent time and for the interesting results, and of course also the people from my group who were involved, especially Christoph Winkler, Matthias Bickel, and Stefan Simic.
Then we move forward, and now it's a pleasure for me actually to introduce Leonard Prince from the University of Padova, and to have him here in this symposium is great. He will talk us, he's a great expert in dissipative self-assembly systems. And today in particular, Leonard will tell us about driving chemical systems away from equilibrium. Leonard, you have 15 minutes, like all the external speakers, plus questions, of course. So please.
Okay, thank you very much, Andres. It's very nice to be here. It also seems that you really had a very nice consortium looking at the research that you've seen. So I will speak a bit about our research interest in this presentation, and it has all to do with, let's say, equilibrium systems. 1 second. Okay, yes, it's going. Basically, we are interested in non-equilibrium systems because life shows us what kind of nice properties you can get from, let's say, non-equilibrium systems. And I think that this is sort of the key essence of what inspires us, is the fact that in a context of ever-increasing entropy, nature is sort of able to create systems that are entropically disfavored. So we are in this entropically disfavored state. And the only reason that we can maintain such a state is because we are able to extract energy from our surroundings. This brings me to the conclusions. The reason why I used these type of mechanisms to illustrate what we're doing is to show connectivity with, let's say, the molecular machine community and the work done on molecular machines. And basically, what we're now seeing is that the same mechanisms that are being used to drive molecular machines, actually we can also apply for the synthesis of molecules. Okay, so clearly that widens the scope. And I put here this reference of a recent review by the Lee group that's really aimed at demonstrating the same concept. Okay, so this brings me to the acknowledgements. I want to thank in particular Tommaso and Ben, who have been very much working on the interpretation of these apparently simple but at the end very complex systems.
Thank you very much. I think it was a nice discussion, and now we go to the last speaker of this session, who is Bob van Sluis from the Radboud University in Nijmegen. He's actually a member of Wilhelm Haag's group, and well, he will tell us about reaction networks.
So today I want to talk about active learning of enzymatic reaction networks in flow, specifically how we can adequately parameterize large kinetic models to control the reactions inside our flow reactors. So we said after 3 iterations, let's see if the model is trained sufficiently well that it can mimic or control the reaction the way nature does it. So we selected different triphosphate ratios, just sampled them, and use the model to predict the conditions that would yield these specific ratios. And long story short, we did find that the model could really accurately predict the actual output ratios, except for the very low concentrations. And that was mainly in the end due to the HPLC, because measuring triphosphates or nucleobases is quite hard on the HPLC. And once we hit the lower concentration regimes, it simply no longer worked. But it took 4 iterations to actually get a model that could do this. And we wondered, is it possible to do it faster? And there's 2 ways. We did 2 test cases. So one test case, we observe more. So instead of using an offline HPLC measurement, we measure online with a timsTOF, but the enzymes are still on beads. And in the other case, we use free enzymes, so we can perturb the free enzymes in the reactor as well to get more information, but we still use the HPLC to measure the output. So in this instance, for the MS measurements, so the online measurements, we coupled 13 enzymes to the glycolysis pathway to beads and flowed the input metabolites into the reactor. And these inputs metabolites or the output of the reactor was subsequently measured online with the timsTOF, which is really nice. Here you see because you can measure a lot more, and time resolution of the measurements are in the milliseconds, or I think, or it's very fast anyway. So here you see 2 different experiments with a different inflow profile, and then on the bottom you see the output as measured by the timsTOF. So we're quite happy with that, and then we decided to see, can we then also extract more information by perturbing more of a network. This was in collaboration with Migle, who had the pentose phosphate pathway enzymes, which were free and allowed to flow into reactors. So we're allowing the algorithm to design inflow profiles for the enzymes as well. In this instance, we did 2 experiments where with experiment 1, we tried to predict experiment 2, and with experiment 2, we tried to predict experiment 1. And as I mentioned, the inflow profile for these enzymes are quite complicated, which allowed us to extract more kinetic data. Long story short, we were able to immediately, after one experiment, after model train on only one experiment, predict the other experiment and vice versa. And this was a long coarse graining procedure of finding different interactions. So, for example, we found that ADP inhibits the PRPPS enzyme, and some of the enzymes had there was a substrate inhibition effect. So it's almost like the enzyme was too crowded if the concentration was too low and the reactivity went down. When in doubt, just perturb your reactor to extract as much information as possible and learn more about the kinetics. With that, I want to acknowledge the people that worked on this, Migle et al. And Wilhelm, and Quentin and Jeroen for the glycolysis pathway. And if you have any questions, then I'm happy to answer them.
So let's start. Welcome back after the coffee break. I hope everyone's revitalized, and I will be hosting the second session of this symposium. And let me introduce our first speaker immediately. We have a pleasure of having Dora Tang here, and just a brief word of introduction. Dora studied and did her PhD at the Imperial College of London, working with Richard Templer on physicochemical studies on lipid membranes. Then for her postdoc, she moved to Bristol to work in the group of Stephen Mann on protocells. And in 2016, she was appointed as a group leader of the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden. And today she will tell us about her group's work on the effect of compartments on reactions.
If we want to be able to build these higher order systems, we really need control and an understanding of how compartments can tune reactions. And actually this is quite non-trivial, because most of our understandings of biochemistry or chemical reactions has really come from taking these reactions and then characterizing them in buffer. And this doesn't really take into account the physical environment that we know that is so prevalent within the cell of compartments and crowding. And so what we've been very interested in as part of our research question is to really understand how compartmentalization can tune reactions as a way for us to be able to kind of control these higher order functionalities within our synthetic cell populations. And in order to do this, we've developed a synthetic cell toolkit, and this gives us a high level of molecular control. And these within our toolkit, we have compartments and reactions with different flavors. We've been using these types of our synthetic cell toolkit and to try to get to this understanding of how compartments change reactions, but ultimately we want to generate these semi-synthetic cell systems which can sustain and drive out of equilibrium behavior, both on the cellular level, but also on the population level. And finally, I'd like to acknowledge my lab, our funding agencies. I think all the people that were involved in the work have been acknowledged in the slides. We do have 2 PhD positions available on a EU-funded ITN, so please contact me if you have any interest there, and I'd be happy to take any questions.
Okay, awesome, great, thanks so much. I think we should move on to the next speaker. Andres de la Escosura, he's originally from Madrid. This is where also he studied and did his PhD. There he worked on supramolecular polymers based on phthalocyanines. Afterwards, he moved to the Netherlands, to Nijmegen, to do a postdoc with Roland Nolte, working on cages and as nano containers, nano reactors, as far as I get this correctly. And in 2009, Andres returned to Madrid, where he grew through the ranks, and since 2018 he's an associate professor at the organic chemistry department, where he leads his group interested in biohybrid materials and systems chemistry. And with that, Andres, the stage is yours.
Saying that I will tell you about programming replication networks and catalysis. Well, we are just starting with that, using minimal nucleobase sequences. So I don't think I have to convince anyone in the audience that, how important is to apply a systems perspective in chemistry. This is, of course, interesting for origins of life research. So we saw a previous lecture precisely about protocell systems. So in principle, it's interesting to implement in chemical systems life-like properties, not only to unravel the origins of life, but also, let's say, there are some technological potentials, which of course we'll not talk about this. It's beyond the scope of this meeting. But anyway, the message here is that if chemists want to go from or mimic the transition from chemistry to biology, we need to do it in the way that biochemistry does, which is basically by establishing, by learning how to deal with very complex dynamic networks with multiple components interacting to each other in different ways, and preferentially, and we have also seen some talks, out of equilibrium, because that's the way to lead to these interesting properties: adaptability, capacity to replicate, that is, to make copies of themselves, to dissipate energy, to maintain the system in a low entropy state, and of course, something which is more difficult to evolve. This is, let's say, the goal. And we need to learn how to deal with network. I think this is a nice system to use the nucleobases to fine-tune the sub-assembly and with the catalytic properties. And for us, is the starting point to try to merge now catalysis and replication in collaboration, and within CLASSY, of course, with Goran Ashkenazi and with Bartosz Lewandowski. And well, I would like just to finish thanking the whole group, of course, and in particular, today I showed work by Sonia Vela, and a little bit in this last part of Alonso Puente. So thank you to them, and thank you for your attention.
Thanks, Andres, again. And let's move on to our next speaker, Silvia Marquesan, and also a brief introduction. Silvia completed her undergraduate in medicinal chemistry at the University of Trieste, and then moved on to the lovely city of Edinburgh for her PhD, where she studied the chemoenzymatic synthesis of GDP azido-mannose derivatives for protein modification. And in 2008, she obtained a fellowship of the Academy of Finland to go to Helsinki to work on integrin biochemical pathways with Carl Gamberg. And then in 2010, for the second postdoc, she went all the way to Australia to work with John Forsyth on biomaterials for regenerative medicine. A few years later, she returned to Italy to the Center of Excellence for Nanostructured Materials, where she also set up her own group, and where in 2018 she became an associate professor in organic chemistry. And Silvia's research focuses on composites of carbon nanostructures, nanomaterials, as well as on assemblies of synthetic short peptides. And I guess it is the latter topic that you will tell us about a bit more today. So Silvia, without taking any more of your time, the floor or the stage is yours.
Yes, we are based at the University of Trieste on the Mediterranean coast, not far from Venice and conveniently near to the synchrotron. And we have two labs, as correctly mentioned. So in one, we deal with composites based on functionalized nanocarbons. In the other one, we deal with the design of minimalistic peptide systems for self-assembly. In both cases, we enjoy working on multidisciplinary projects, and we like to develop functional nanostructures, especially in green solvents, so water and acetonitrile. Started to use these systems in very simple proof of concept examples that show you can use them and apply them for catalysis. And this encompasses examples in biocatalysis, so typical enzyme mimicry, but also organocatalysis. We've seen beautiful examples earlier on today with proline, but also I think Rink Elkema showed some thiourea, if I remember correctly. So you can use your creativity there, and photocatalysis through collaborative works mainly. Bear with me. Here is the acknowledgement slide. This is our group. It's a very young group. There's loads of master students. In terms of collaborators, I'm indebted to the biophysicist, now actually Professor Attilio Vargio at the University of Cagliari, who with all atom molecular dynamics helps us understand how these simple peptides self-assemble into more or less complex structures. There's loads of crystallography that we do, thanks to the help of various crystallographers at the University of Salerno and also at our institution. For the photocatalysis, Luca Dell'Amico is expert in the benzophenone type photocatalysis. Funding mainly from the Italian Ministry of University of Research. I'm indebted actually also to the Royal Society of Chemistry, because when our lab was penniless, they provided us with some basic funding that enabled our group to stay alive. So I'll be forever grateful to the RSC for that. And lastly, there is an advertisement slide. We are organizing an international conference, ISNSC 2024, that aims at bringing together the supramolecular and the nano communities. So I hope this will be of interest to some of you. It will be held at the beginning of October in a terrific location that is the southern coast of Sardinia, where in early October, it's typically sunny and warm enough to swim in the Mediterranean.
It was great having you here. So with this, we move on to our last speaker of the day. And this is again one of our wonderful CLASSY partners, Gonen Ashkenazi. And then again, let me give him a brief introduction. Gonen carried out his master thesis as well as his PhD studies at the Weizmann Institute, where he worked with Professor Abraham Schanze on supramolecular assemblies based on metalloporphyrins. And after completing his PhD, he in 2001 went to the US, to California, to the Scripps Research Institute as a Fulbright Fellow, where he worked as a postdoc with Reza Gadiri. And there he studied dynamic molecular networks based on synthetic peptides. And in 2006, he returned to Israel and joined Ben Gurion University, where he still works until today. There, he quickly rose through the ranks, and in 2015, he became a professor of chemistry. And the main research focus of Gonen's group, I would say, is multi-component chemical systems and molecular networks. And today, he will tell us a little bit about his bistable and oscillatory behavior studies in replication networks out of equilibrium. So, Gonen, the stage is yours.
So, very briefly, Andres showed the experimental system that we work together on for the replication of nucleopeptide systems, and this system the molecules can replicate individually, and they can also do cross-catalysis, and they can form a simple network where there is some selectivity towards one of the replicators. Under CLASSY, we worked on our role under CLASSY was nucleic acid peptide and nucleopeptide system. So we worked on this system and also on another analogous system, but this will not be the topic of my talk today. I think that one of the common themes from all the lectures we had so far is trying to mimic biological complexity. So we try to do this with peptide networks. For example, we try to mimic the autonomy of the system, to try to see if we can get spatial and temporal self-organization, heredity aspects of replication, reproduction, selection in the mixture, and also metabolism. But actually, what I'm going to describe is a modest approach. So we are not trying to reproduce a zebra yet, but we try to do something that is much simpler. So biosignaling in the most simplest way, we can think about this as a small molecule binding to a receptor, which starts a downstream process to activate different reaction networks that are important for the cell cycle or the cell behavior, the cell response to different triggers. And the two systems that one can think about, one is the bistable switches that are involved in cell division. Bistability means that depending on the exposure to different triggers, the system will reach one of two steady state, and this steady state can be considered as a fixed trigger, fixed input for downstream behavior. And the second system is based on the core oscillator of the cyanobacteria, which is made of three enzymes, the Chi-A, Chi-B, Chi-C, and it was shown that this simple ternary system, if we combine it with ATP, can oscillate with respect to phosphorylation of one of the enzyme of the CLASSY. So in our system, we try to use peptide networks to demonstrate or to reconstitute bistable motifs and oscillatory motifs. When we provide all the ingredients I discussed before, the system will oscillate. Of course, we can say that these oscillations are far from perfect, but we worked on this by changing the conditions, for example, by lowering the flow rate, and we were able to get oscillations that look somewhat more robust, more consistent. After the first burst, the rest of the oscillations are consistent. So this was a very fast tour on the one aspect of the research in my lab, which was not studied, which was not directly studied under the framework of CLASSY, and I showed two different examples. One is that if we use reversible replication, we can get a bistable system, and we can use this bistable system to form switches. And if we take the irreversible system and studied it in the under conditions of flow and under appropriate configuration, we can get the system to oscillate. I want to finish my lecture by thanking the students and co-workers in my group. Those are mentioned in red, are part of the projects that I showed so far. I want to thank our collaborators, of course, especially Andres de la Escosura, that also was coordinating the CLASSY. I want to thank all the CLASSY partners, PIs, and students for excellent program, which is now over in 1 month. So we will meet in other occasions. But anyway, thank you very much, and thank you all. I'm happy to take questions.
Thank you, Gonen, once again, and all the speakers of today for their contributions, for really, really interesting talks. I thought this was really, really excellent. And before we finish, I'm gonna give the microphone back to Andres. I guess he wants to say a few concluding remarks.
Thank you all for being here, especially the invited speakers and the CLASSY members also. This last discussion was very interesting. Today we saw many things of out of equilibrium, self-assembly, catalysis in reaction networks, but there are different ways to approach, for example, out of equilibrium, this discussion, we can understand different things. So there are different ways to reach out of equilibrium systems. So I hope the meeting was useful to all of you for getting a little bit more in depth into these complex issues. We are all dealing with complex systems, and well, for me it was a pleasure.
Okay.